Good morning everyone. How are you students? Everyone fine? So in last class we were discussing about reflection. Okay, and specifically we discussed about reflection by spherical mirror. We discussed two type of mirror, concave and convex. Okay, and we understood sign convention also. What is the use of sign convention? For any mirror, incident ray direction is positive. If object is placed here, so from the object ray will incident like this. So this direction positive and this direction negative, above it positive, below it negative. Okay, this point we discuss. Today we will use this concept. Okay, so we will use this concept in mirror formula. So here we will not derive the formula directly, we will use the formula. About focus, everything we discuss. For concave, what you have to remember, for concave mirror, focus is this side, right? Focus is this side, है reflecting side. Okay, and for convex mirror, focus is this side, है not towards reflect. This is reflecting side. This is silvered side, so this focus is towards silvered side. Okay, now what is focal length? The distance of focus from the pole is known as focal length. Distance of focus from the pole is known as focal length. Okay, so this distance, here we can draw this distance. This is focus, this is pole, so distance between them is focal length. We represent it by f. So what is f? F is focal length. F is focal length. Focal length means distance between pole and focus. Okay, now students, answer me this point. Focal length is positive for which mirror, negative for which mirror? Can you answer this question? Focal length is positive for which mirror and negative for which mirror? Very simple. What I said earlier, we discussed sign convention. Incident ray direction positive means this direction is positive and this direction is negative. Okay, so here this is the position of focus. Now everything we have to calculate from pole. Already we discussed this point, that all measurement we have to take from pole. Okay, distance are measured from the pole of the mirror. So here also, if we have to measure focal length, so we have to move from pole. So this is pole, this is focus. How we are moving? Pole to focus in this direction, right? And already we discussed that incident ray direction is positive. Ray is incidenting like this, means this direction is positive and opposite to it, this direction is negative. This is negative, this is positive. Okay, so here we are moving from pole to focus in this direction, so this direction is negative. Therefore focal length is negative for concave mirror. Okay, so focal length, you have to remember this point. In next slide I will write it, that focal length is negative for concave, this side negative. Now understand for convex. Here is the pole, here is the focus. We are moving pole to focus in this direction. So this direction is positive. Why? Because rays are incidenting like this. So this direction is positive and this direction is negative, right? So pole to focus, this direction negative, sorry, positive. So focal length is pole to focus, focal length is positive for convex. Is it clear? So positive for convex and negative for concave. So next point that we have to write here about focal length. And here also in all these diagram we can understand, this is which mirror? Concave. This is focus, right? Okay, focus is here. They mention focus is here. Now, pole to focus. Rays are incidenting like this. This direction positive, this direction negative. So, pole to focus negative. Okay, here negative. Here we can see this rays are incidenting like this. So, this direction positive, this is pole, this is focus. Pole to focus positive. So, focal length positive. Focal length positive for convex mirror. Okay, and here we can see opposite, so it is negative, right? So focal length negative for concave mirror. Okay. Now next point we have to understand about real object, virtual object, real image, virtual image. Okay. So first point, what is object? Real, virtual, that is different thing, but first we have to understand what is object. So you all are real object. Means what? Whatever we see in really, if anything exists, so that is what real object means. Bottle, here I can see bottle, laptop, okay, this is screen, LED screen, light, fan, all these are real object. What is the meaning of real object in physics language? In physics language, real object means, understands, rays are incidenting on this object. After then rays are first incidenting, then reflecting. Incidenting and reflecting. We can see this hand from all side, this side, anywhere, right? Means in all direction this object is reflecting the rays. In all direction this object is reflecting the rays. Means real object means what? That rays diverge from the real object. Is it clear? Rays diverge from the real object. Those rays only incident at our eyes, that's why we can see that object. Okay, so real object means, real object definition is, and we can see here virtual object, real object. Okay, so here they taken real object, so you can see here this is real object. Now rays are diverging from it. First ray this one, okay, second ray this one, rays are diverging. Agree or not? Okay, so this is what? Real object. So real object means rays diverge from it. Okay, now understand, this is real object, we understood. Virtual object. So on this mirror rays are incidenting. Okay, from where these rays are coming? Can you say their origin, source of origin, from where they are coming? We can't give answer. Why? In this we can give answer. How? If anyone asks you that from where this ray is coming, so we can extend this ray. Okay, we can extend it. They are intersecting here, means we can say they are coming from here, right? So we know actual position of from where rays are coming, from where rays are originating. We know the actual point, right or not? That is real object. But here if we extend all these rays, these two rays, are they intersecting anywhere? No, they are not intersecting. If they are not intersecting, means it is not real object. Rays are incidenting means it is related to object. Incidenting ray means related to object. This is also important point. Incident incident rays means related to object. Now it is real or virtual. How to find? If incidenting rays are intersecting, means real. If they are not intersecting, means it is virtual. So here we can see these incidenting rays are intersecting here, so it is real. But here these incidenting rays are not intersecting, right or not? That's why it is what? Virtual. So virtual object means we can say that these rays will intersect where? Nowhere. Nowhere means what? Infinity. So yesterday I did not use infinity word. Now today I am using it. So what is the meaning of infinity? Again listen. Where these rays will intersect? So these rays will not intersect. Nowhere. Okay. Nowhere means if anything does not exist, that is what? Infinity. Is it clear? What is infinity? Here infinity meaning is they will never intersect, means it is they will not, intersection point is not exist. If it is not exist means where it is at infinity. Infinity means which does not exist. Is it clear? So we give name that these rays are coming from infinity. Is it clear? And both here they taken like this, but we can use these parallel ray also. If we extend these parallel ray back where they are intersecting, nowhere. Nowhere means we can say from where they are coming, infinity. Is it clear? So infinity you understood? Okay. Now next, virtual object means where incident rays does not intersect. Okay, but we can give here different type of definition. Understand this point. So in reality these rays will not intersect, but understand, if we extend this ray, actual path, understand this point very carefully, actual path of this ray is only till this point. Why? Because here mirror is there, and after this point this ray will reflect, right or not? So this ray reflected after this point. This ray is not traveling in this direction, in this side. Agree or not? Okay, but we are extending it. Okay, now if we extend both rays, then they are intersecting here. So are they really intersecting here? No, they are not really intersecting here. So what we use word here? Seems. Both rays are seems to meet. Seems to meet where? At virtual object. Is it clear? Rays, if rays are seems to meet, that is virtual object. I said you that this ray already reflected here. This is not the path of the ray. We extended, so it seems, because if any person is this side, if any person is this side, okay, so what that person will think if this mirror is not visible? That person will think that these rays will meet here, right or not? But a mirror is there, so before meeting this point they will reflect. Okay, so in reality they are not meeting. So we will use here this word that it seems to meet at a point. That point is virtual object. So this is the position of virtual object. So what is the definition of virtual object? Incident rays are seems to meet. Okay, so virtual object definition is incident rays are seems to meet. Okay, understood? So we understood both real object and virtual object. Now understand image. So first point, How image will form? How image will form? So image is formed by reflected ray. In mirror, what we see? Our image. Agree or not? So what mirror is doing? Reflection. Right or not? Reflection of light. So that image is formed by reflection. Okay. Means image is related to reflected rays. Okay. The object is related to incident rays and image is related to reflected rays. So here image related to reflected rays. Okay. Now real image, virtual image. Virtual image and real image sense is what? To understand this example. Okay. Here is the object. This is real object, yes, because rays are diverging from it, yes. After then these rays reflected like this. Okay, both reflected rays are intersecting? No, they are not intersecting. If they are not intersecting means if they are not intersecting means they will not form real image. Is it clear? They will not form real image. Okay, now if we take one more example here. Okay, this example. Understand this point. Here is the object. This is what? Object. Okay, how to identify object? These are incident ray. Okay, these incident rays are diverging from object. Yes. Okay, rays are incidenting like this. Yes. Okay, now here reflection, because mirror is there, so this ray reflected, incidenting, this ray reflected. Okay, now this is reflected ray, है this is reflected ray, this is also reflected ray. Both reflected ray are intersecting here. Agree or not? So both reflected ray are intersecting here means this is the position of real image. So this is the position of real image. So this is what? Real image. Is it clear? So real object, this is real object. We can see that rays are diverging. Okay. And rays are, if reflected rays are intersecting, that is real image. If reflected rays are not intersecting, that is virtual image. Is it clear? Okay. So we understood this point. Real image means if reflected rays are really intersect, to real image. Okay. We can see this one more example. This is object. Rays are incidenting on the mirror. This is reflected. This is reflected. Both are intersecting here. Both reflected rays are intersecting here. So this is real image. And this is what? This is real object. Okay, so we understood real image. Real image means if reflected rays are intersecting, real image. Real image. Real image means reflected rays intersect at a point, that is real image position. Okay, so we understood real image. Now next is virtual image. So here we can see these are reflected rays. Okay, these reflected rays are not intersecting. Now if we extend them back, so this is not actual path of the ray. Actual path is, this is actual path, है ना? This is actual path, but this is not actual path. This is actual path, but this is not actual path. But if we extend them, so it seems that these rays are coming from here, right or not? So this is the position of image. Which image? Virtual image, right or not? Why? Because it is behind the mirror. Behind the mirror, nothing is there. Behind the mirror, nothing is there. You know, in plain mirror, every day we see our image. So we see our image in the mirror. Okay. If we move away, so our image also move away, right or not? But so we see our image in the mirror. So anything is there in the mirror? No. And behind the mirror, are you there? No. So that is what virtual image, right or not? So here this is virtual image. So virtual image means reflected rays really does not intersect. Okay, first point. Second point, if we extend them, then they are seems to meet at a point that is real image, that is virtual image. So this is virtual image. Reflected ray, here I will write the definition, and this is also virtual image. This is also virtual image. Okay. Now virtual image definition is reflected rays seems to diverge. Seems to diverge. From where these rays are coming, it seems that they are coming from this point. So seems to diverge. Clear? Okay. So we understood real object, virtual object, real image, virtual image. All these points we discuss. Okay. Now next is, and here you can see real object image can be real object image can be virtual, and real object image virtual object, this is virtual object, rays are incidenting, virtual object image real, virtual object virtual image also possible. You can see how rays are incidenting, these are incidenting rays. So if we extend back, are they intersecting? No, means it is virtual object. How they reflected? They reflected like this. So this is reflected ray. Are they intersecting? They are not intersecting. Okay. So this is which image they will form? Virtual. So virtual object, virtual image. So all case we discuss. Now we will understand about mirror formula, mirror equation. Okay. Mirror equation. So all you are aware about it. Mirror equation, mirror formula is 1 by V plus 1 by U equal to 1 by F. You know it. Okay. What is V here? V is image distance. And you know all distance are from pole. And what is U here? Object distance. This is also from pole. Okay. And what is F here? Distance of focus. Already we discussed. We call it focal length. Okay. Now by using this equation, we can calculate image distance from the pole. Okay. If object distance we know, we can calculate image distance. Okay. Now we will solve one example. And in this formula, we have to put all value with sign convention, right? Sign convention we discussed now. Okay. And here one more point we have to understand, magnification. Magnification means height of image divided by height of object. Okay, and its formula is, we represent it by m, and its formula is minus v by u. This is the formula of magnification. Okay, one more point here you have to remember: erect image and inverted image. Erect image means object and image both are same side. Erect image means object and image both are same side. We will use all these points in question solving. And inverted image. Inverted image means object and its image both are opposite side. Okay, so we understood about erect image, inverted image, magnification formula and mirror formula. Now we will use all these in questions. So one example. Okay, solution is there, no need to worry, just understand, because after solving it we will solve one question also. So what is given here? An object is placed at a distance of 15 centimeter. So first you have to know what is given. So what is given? Object is placed at a distance. This is object distance. Object distance means u. So this is what u. We will write here only that this is u is given. Okay. In front of a concave mirror. So mirror is concave. Now at same time you have to think where is the focus. Focus of concave mirror this side. Okay, and focal length is negative. So focal length also given, means what is given? F is given. It is negative, so we will use negative sign. Okay. Describe the image formed and calculate the distance from the image. So calculate the distance from the mirror means what we have to find out? V. We have to find out V. Okay, so we have to calculate here V value. This V we don't know. What is given? U given, F given, V we have to find out. So mirror formula we will use. So mirror formula we all know that 1 by V plus 1 by U equal to 1 by F. Now understand how to put values. Okay. So actually where is the object? In front of the mirror. This is mirror. Here is the, we can locate the object also, but no need. Just only you have to know that where is the object. This is the mirror and this object is here. Okay. So object is this side. Do you know from the object how ray will incident? Ray will incident like this, right? These are incidenting rays. So rays are incidenting like this. If rays are incidenting like this, then this direction is positive and this direction is negative. Agree or not? Now object distance from where we will measure? From pole. This is pole, this is object. So pole to object we are moving in this direction and this direction is negative. So object distance is negative, not positive. So U value 15, okay, but sign we have to use. Which sign here? Negative sign, because this direction negative. So U value is minus 15. Is it clear? Object distance they given, but you have to put these values with sign in the formula. That's why here you have to use sign. So minus sign, because this direction negative. Now focal length, already we discussed focal length of concave mirror is negative. Why? Because focus is this side, focus is this side. So again focus this side, so this is the position of pole, pole to focus, this direction negative. So focal length is negative. Okay, so we will put these values. We have to find out. F value we know, U value we know. Put the values and solve it. So finally we got here answer that Image distance is how much? -30. Now understand this point. If anyone asks that which side this image, this side or this side, okay, तो V negative है ना, image distance negative, negative means this direction, right or not? So image will form this side. Is it clear? Now understand this point, very important point, how to find that it is real image or virtual image. Very important point, how to find it is real image or virtual image. Do you know real image means if reflected rays cut, then it is real image. So reflected ray can cut which side, this side or this side? So you will say definitely sir, reflected ray can cut this side because this ray will reflect. So after reflection it will move back in same medium, this side only, right or not? So reflected ray can cut this side. Okay, so if image distance negative means image is this side, if image is this side means this is the position where reflected rays are really intersecting, means it is what? Real image. If V value suppose positive, suppose V value positive, if V value positive means image is this side, and you know this side reflected ray can't travel, right or not? Means virtual image seems to meet, they will seems to diverge from that point. So this side virtual image, this side real image. To this point also you have to remember what important point it is, how to find that it is real image or virtual. So this is concave mirror. If image is this side, so this side image are virtual. If image is this side image are real. Okay, this is virtual image side. If image form V value positive means virtual image, V value negative means real image. This side real image, real image means V value negative. Okay, and virtual image V value positive. Okay, now convex. This is convex mirror. Again reflected ray side, this side image are real, this side image are virtual. So this side virtual image, this side real image, V value negative, V value positive. Okay, so we understood everything. Magnification also we can calculate here. Magnification formula minus V by U. Everything you have to put with sign. V value how much we got? -30. U value how much? -15. Put here. Finally we got negative. Okay, clear? Now one question for you. This question you have to solve, and let's see that how many of you understood this topic. You have to find out this question answer. 4 options are given. Which option is correct? Option only you have to send. An object is placed at a distance 10 cm from the focus of a concave mirror. Remember which mirror is given. And image is at a distance, image position also given here, 40 cm. This is 40 cm from focus. The focal length of the mirror, we have to find out focal length of the mirror to calculate. Focal length value is how much? Focal length value is how much? Now solve this question. Let's check that which one is correct. So which mirror it is? It is concave. But focal length we have to find out. Focal length we have to find out. Focal length they did not give. So no need to worry that focal length sign we have to put, right? Because we don't know about focal length. Okay, so this F we have to find out. Now V and U. Object distance 10 centimeter from the focus. Okay, so one more point here, interesting point, that object is placed at 10 centimeter from the focus. They did not give from the pole. Interesting now. Twist is there in the question. I think you did mistake here. This distance is given focus, okay. And image distance also given from the focus, right or not? Both are. Here this image distance is also given from the focus. Okay, so both are given from the focus, not from the pole. Is it clear? So we have to find out, and in this formula we have to put all these distance from the pole, right or not? So we have to add focal length in these distance, then we will get answer. I think you directly put here distance value, V value 40 and U value 10, and you solved it. Okay, so here in this question, this is focus, okay. So from the focus, object distance is 10 centimeter. This is object, and you know this is F. We have to take U value, means distance of object from the pole. So U value is how much? It is 10 plus F. But in this formula we have to put negative, right? Because from the object ray will incident like this direction is positive and this direction is negative. Now image distance also given from focus. How much distance? 40. So 40, obviously it is more, so image must be this side because distance is more, and this distance is how much? 40 from the focus. So from the pole how much? 40 plus F. So V value is 40 plus F. And here also negative. Why? Because this direction negative, this direction positive. Agree or not? Now put all the values. So 1 by V value is minus 40 plus F, and U value also negative. So if it is negative, so here it will become negative. 10 plus F equal to 1 by F. So this equation we have to solve. Understood? Is it clear? Okay. To solve this equation, then you will get answer. Understood all of you? Okay. So your answer can be, I think, this third option. Next class is there. So this third option or this second option. Out of these two options, any one, because 10 and 40 are not possible. If you will put here 10, so just by option we can check. F value is, and you know this is concave mirror, so focal length must be negative. So minus 10 and plus 10, 0. So 1 by 0, infinity. It will become infinity, right? We are unable to solve. And if we put 40 here and focal length negative, okay, so minus 40 and plus 40, again 0, 1 by 0, infinity, right or not? So these two options 10 and 40 are not possible. By option also we can check. Put here minus 20, then if we are getting equal both sides, that is our answer. Put here minus 30, if we are getting both sides equal, that is our answer. So these are not possible. Put 20, if we are getting equal, that is your answer. If we are not getting, then answer is 30. Is it clear? So we have to solve this equation. So you all, okay, now you are sending second option, okay, means that is correct. Okay, very good. Okay students, so we understood this mirror formula and we solved question also, and you all done mistake Why? Because you directly put here 10 and 40, but here in the question it is clearly written that these distance are given from focus. Is it clear? Okay, students. Further we will discuss in next class. So I hope you all enjoyed this session and you understood the concepts. Thank you.
Good morning everyone. So yesterday we have seen balancing redox reaction. What are the various steps we have seen? First, we need to find oxidation states and figure out which element is changing the oxidation numbers. This is the first. Second one, calculate the total change, take the ratio of it and cross multiply. And finally, third step is balance the other elements which are not balanced, balance them normally only. Now, yesterday we have seen few examples. Let's see one more. Fe2O3 plus CO gives Fe plus CO2. Now let's balance this using that method which we discussed yesterday. Now what are the oxidation states? Normally oxidation states, oxide means -2 we will put. So, and whenever elements are there, I mean if atoms are not connected with any different atoms, then it is elementary state, elements always 0. And if oxygen is -2, carbon will be +2, then only overall will be equal to the 0. If oxygen is -2, then iron will be how much? Let's take X and find out that. So 2 irons are there, 2 into X plus 3 oxygens. 3 into -2 is equal to overall charge. Overall charge is what? It is not an ion, so 0. So you will get X is equal to +3. So oxidation state of iron is what? +3. Now, which are undergoing change? Iron is undergoing change. And what else is undergoing change? Here carbon also. Oxygen -2 means carbon you will get +4. So carbon is also undergoing change. Find the total change. Total change, first see, yesterday I mentioned that first point is actually balance the elements which are undergoing the change. Yesterday's example already they are balanced, so first step we did not utilize there. But see here, which are elements undergoing change? Iron, carbon. Are they balanced on left and right? Yes, one carbon, so no problem. But iron, if you see, here one iron is there, but here 2 irons are there. So iron is not balanced. So just balance it. Just how to balance? Just put the number 2 here. That's it. So elements which are undergoing change are balanced. Now calculate the total change, ratio and cross multiply. So what is the total change? For one iron atom, +3 to 0. For one iron atom, what is the change? +3 to 0 means change is what? Positive change 3 for one iron atom. But how many iron atoms you see here on that picture? You see 2 iron atoms. For one iron atom's change is 3. For 2 iron atoms, how much? So total change is equal to 2 iron atoms. Each iron atoms, what is the change? Positive change 3. So total 6. Okay. Now come to carbon. Carbon, what is the change? +2 to +4 means what is the positive change? 2. So, and carbon atoms, number of carbon atoms also only 1 only. So total change is 1 carbon is there and its change is 2. So 2 is the change. Now take the ratio and cross multiply. So iron, carbon, what is the ratio? 6 is to 2. Means simplest ratio is what? 3 is to 1. Now cross multiply iron substances with 1, carbon substances with 3. 1, we multiply with 1 means nothing will change. Already 1 is there in the picture already. So therefore nothing is there means 1 is there already. 1 into 1 means 1 only. No need to change anything. So I will rewrite this again first. Fe2O3 plus CO gives 2Fe plus CO2. Now, so iron 1 you have to multiply 1 into 1 1, Already 2 is there, 2 into 1, 2. So don't change anything. Now carbon multiply with 3. So already 1 is there before CO, carbon substance. 1 into 3 is 3. 1 into 3 is 3. So this is the balanced equation of this reaction. Is it understood now, everyone? Reply. Okay, good. Now let's see some problems on this. Okay, before going into problems, I will discuss one more case, which is also very important. You will learn this again once you come back when official classes are started. You will learn this in the stoichiometry chapter again. If you understand now, then you will understand better there at that time. You will be faster at that time in calculating things and answering the questions. So, but this we will learn again afterwards, don't worry. Now, we have seen how to balancing. We see one more case in this balancing reactions when ionic equation is given. Here we have seen 3 examples. I studied 2 examples, now 1 example. In all these 3 examples, there is no ion is involved on the reactant side or product side. All are compounds only. Suppose if ions are involved, then almost procedure is similar, only one step extra will be there in case of ionic equation. Okay, see what is that. We will learn that and then we will go into the problems. Now see, we are discussing ionic equations, how to balance. Suppose I have MnO4 minus plus Fe plus 2 gives Mn plus 2, H plus gives Mn plus 2 plus Fe plus 3. Suppose there is an equation like this. Then how to balance this? Actually, sometimes they will give water, sometimes they don't give. That you have to use finally to balancing, for balancing purpose if required. We will use that anyway. So sometimes they will give that, sometimes they don't. It's not compulsory they have to give. We can utilize that at any point. Okay. Now how to balance? First step is what? Say first step in the earlier we have seen, first write the oxidation states and see which are undergoing change. Balance them first. If they are not balanced, that is the first thing. So let's find the oxidation states. When you find oxidation states, oxygen minus 2, first let's put oxygen minus 2, hydrogen plus 1, here already visible only in the ion H plus 1 means plus 1, iron plus 2, here for ion element, if only single atom is there, okay, single element is involved in ion, so its charge will be its oxidation state, so plus 3, and manganese also plus 2. So here manganese is what? Let's take it as X and figure out that X. You should get plus 7 there. See? Yeah, 1 manganese. So 1 into X, 4 oxygens. 4 into minus 2 is equal to overall charge. What is the overall charge of this ion subspecies? Minus 1. So if you calculate that, you will get plus 7. So manganese oxidation state plus 7. Okay. Now which elements are undergoing change as you see here? Which elements are undergoing change in oxidation state? Write it in the chart and tell me. Yes, manganese is changing as well as iron is changing. So manganese, iron. Okay, fine. Now see whether they are balanced or not, left and right. 1 manganese here left side, 1 manganese right side, balanced. 1 iron left side, 1 iron right side, they are also balanced. So we need not bother about balancing them, already they are. Next is find the total change. Total change is what? Here 7 plus 2, 7 minus 2 is what? 5. So total change here 5. Here total change is what? 2, 3. Positive change is what here? 3 minus 2 is 1. So ratio is 5:1. Means manganese is to iron. Change is what? Ratio 5:1. That is already simplest ratio. Now cross multiply. Manganese items you have to multiply with 1. Iron items you have to multiply with 5. Okay, whichever is multiplied need to be multiplied with 1, you need not bother. Then no numbers will change there when you multiply with 1. So focus on iron substances. We have to multiply with 5. So I will write the equation again. MnO4 minus Fe plus 2 H plus Mn plus 2 Fe plus 3 water. Okay, now what we have to multiply? Iron items you have to multiply with 5. Where is the iron items? Here iron is there. Nothing is there means initially 1 is there. 1 into 5, 5. 1 into 5, 5. Okay, done. Now up to here what we have done, what is actually happened by this time, when we cross multiply, we are balancing the electrons involved in oxidation equal to the electrons involved in reduction. We achieved that at this point. Okay, remember that. Okay, now after actually in the earlier procedure what I taught yesterday, after this cross multiplication, what is the next step? Balance the elements which are not balanced. If any other elements are there, balance them. That is the step I taught yesterday. But now in ionic equation, one more step will be there. Before the last step, balancing the other elements, one more step will be there in the ionic equation. What is that? Now, charge balance. That step is, remember this step will involve only in ionic equation when ions are there. If it is all neutral compounds, then no need of this step. Already charge left side LHS will be 0 charge, right hand side 0 charge. 0 is equal to 0, already charge is balanced. But in the ionic equations only this step we have to take care. Now charge balance is what? See, left side if you see, I will take LHS, left hand side, RHS, right hand side. Let's see left hand side what is the charge, total charge, and right hand side total charge. If they are balanced, we are okay with it. Not balanced, then not okay. We have to make it balanced. Now, so what is the charge here? Permanganate. Here see 1 MnO4 is there with charge -1. So what is the overall negative charge here? -1. 1 into -1, total negative charge -1. Iron, positive charge. How much is there? 1 iron has +2. Like that 5 are there. 5 into +2. What is the total charge? 10 plus 10. Okay, this is the total charge. And don't take this H plus into picture. Don't calculate its charge because this is our trump card. Using this H plus only we will balance the charges on both sides, because so far the number before H plus is not fixed. H plus we have not taken into account in any of the calculation. So number before H plus is not fixed. What numbers are fixed? Before the manganese items, before the iron items, numbers are fixed. So focus there only. Don't take the H plus into account. Okay. Using this H plus only we will finally balance the charges. Okay. So left side then overall charge what we got by using these two items? -1 plus 10 means how much this is? +9. Okay. Now right side. 1 manganese is there, its charge +2. 1 iron charge +3. 5 means what? 15. 5 into 3, 15. Don't take water into consideration. Now, so now total charge on right side is how much? 17. Left side 9. Are they balanced? No. We have to balance. How to balance? So listen carefully. If you are taking the help of H plus ion to balance the charge, in this case we have to use H plus only to balance the charges. So if you are using H plus, means acid medium, to balance the charges, then add that H plus, required number of H plus, to the lower charge side. See, there are two ions, H plus, OH minus. Either we use H plus to balance the charge or OH minus to balance the charge. Okay. They will mention in the question, they will give a hint in the question which ion we should take support to balance the charge. In this already we got idea which ion we should use to balance H plus. Sometimes they will write reaction takes place in basic medium, like that they will give. From that we get hint we have to use OH minus to balance. Okay. Now if you are H plus OH minus, suppose if you are using H plus support to balance the charges, now plus H plus OH minus, plus 1 minus 1, plus is bigger number or minus is bigger number, you remember like this so that you don't make any mistake. Plus is bigger number or minus plus is bigger number. So H plus if you are using, add it to the lower charge side to achieve the balance. If you are using the support of OH minus, minus means a smaller number, so smaller value generally numericals if you see, then add this OH minus if you are taking its support to the higher charge side. This is the hint. Okay. H plus if you are using lower side you add, OH minus higher side you add. Now see, we are using the support of which one? H plus. So we have to add it to the, plus means bigger number, right? We have to add it to the lower charge side. Which is lower? Here 9 number is there, here 17 number is there. Which is the lower charge among these two? 9. So this side we have to add H plus. How much we have to add? Whatever difference is there, difference in charge, that much we have to add, because we are achieving the charge balance. So what is the difference between 9 and 17? 9 and 17 difference is what? Add H plus on lower charge side, add H plus on the lower charge side. So what is the value of H plus? Number of H plus to be added is equal to what? Difference in charge. What is the difference in charge? Positive difference you take. 17 minus 9 is how much? 17 minus 9? 8. So 8 H plus we have to add. Okay. So I hope up to this clear. Now I am going to the next page and write the equation again so that I will show you. So MnO4 minus 5Fe plus 2 plus H plus. How many H plus we have to add? 8 H plus we have to add. And Mn plus 2, 5Fe plus 3 and H2O. H2 is one more thing remaining, right? After this, then last step. Last step is what? Balance the elements, other elements which are not balanced. Here, what are the other elements besides the manganese and iron? Oxygen and hydrogen. Balance them normally by looking at them. So if you see 8 hydrogens are there left side, but right side 2 hydrogens. So you have to make it 8 by putting 4 here. Automatically other element, remaining oxygen element automatically get balanced. You need not bother about it. Okay. See, if you check here how many oxygens? 4. Right side also see 4 oxygens are there, balanced. This is the ionic equation. Is it understood, everyone? So now let's see few problems. Balance the following half reaction for the unbalanced whole reaction. Okay. See, there is a reaction given CrO4 minus 2, CrO2 minus OH minus. We have to balance this and we have to select the right option among them. Okay. Now here it is, what is the half reaction? Half reaction means it is not complete reaction. Means it is not oxidation and reduction, both are not involved. Either oxidation or reduction only involved in this equation. Then we should call it half reaction. If both oxidation and reduction involved, we call it full chemical reaction, but it is only half. Okay. Now let's see, here chromium oxidation state if you find +6 you will get, and here if you find +3 you will get. You know how to find by taking X value, and I already showed you. If you do it, you will get like this. and oxygen minus 2, hydrogen plus 1. Now, and here also oxygen minus 2 only. So oxygen, hydrogen usually they don't change. So focus on other element. So chromium going from plus 6 to plus 3. So how to balance this one? Now see, when you are balancing the half reactions, we have to balance with electrons, because in full reaction, the electrons involved in oxidation and reduction, they get cancelled. That's why we don't see any presence of electrons in the full reaction. But only half reaction is given, then electrons must be there in the equation. How much, what is the change in oxidation state is there, corresponding electrons must be there when you are writing a half equation. So here what is the change happening? One chromium is only involved, and further change is 3. Always oxidation number change happens due to electrons only. So 6 to 3 means change is 3. So 3 electrons are involved. Now which side I have to add electrons, sir? See, left side I have to add electrons, or right side I have to add electrons, 3 electrons. Now see, tell me, electrons have negative state or positive? Negative. Electrons have negative charge. Okay. So negative charge, if we add, suppose if we add electrons to the right side. Okay, suppose left side, what is the charge is there? What is the oxidation state? A charge plus 6. Right side 3. If you are adding electrons, 3 electrons. 3 electrons means you are adding minus 3. Because electrons have negative charge, minus 3. If you add minus 3 on the right side, it will become 0. Balance will not be achieved. But if we add electrons to the left side, so electrons means 3 electrons means minus 3 charge, 6 minus 3, 3. Therefore, always add electrons to the higher oxidation state side to balance the oxidation states. So left side you have to add 3 electrons. So see which option is having the electrons on the left side. Among these, if you see, electrons are, first of all, electrons are present here and here only. And among them, 3 electrons are present on the left side. This is the answer. Rest other 2 options you need not bother, because electrons are not there in that. Half reaction means always electrons has to be there. Next, we will see next question. There is a reaction given. What is the value of N? N means electrons. This is also half reaction only. So just now we have seen a previous example. Similarly, we will find out. So manganese, what is the oxidation state? See, MnO4 minus is going to Mn plus 2 state. What is the oxidation state? If you find plus 7, you will get plus 2. So what is the change? 5. Therefore, 5 electrons will be involved. Which side I have to add? Obviously, in the question they have shown on the left side, but always higher side you have to add. So 5 electrons. What is the answer then? First one. 5 electrons. Correct, correct. Next. Okay, look at this. For the redox reaction, zinc and nitrate gives Zn plus 2. In basic medium, see what is given? Basic medium. It is an ionic equation. See, ions are there. Ionic equation. Basic medium means for charge balance, which support you have to use? H plus or OH minus? OH minus. OH minus support you have to use in balancing. Okay. So they are asking what are the coefficients? Coefficients means the numbers. The numbers before the species we write now in the balancing. 2 Zn, 3 Zn, like that we write now. Those numbers are called coefficients, stoichiometric coefficients. So we have to find the coefficients in the balanced equation. Okay. Now let's take Zn, NO3, Zn plus 2, NH4 plus. Okay. Now elementary state. Elementary state means oxidation state is 0. Oxidation state is 0. Nitrogen. Take oxygen minus 2 and find the nitrogen oxidation state by taking it as X. Okay, I will show you here. but others you find out on your own. So oxygen minus 2, nitrogen X. So 1 nitrogen 1 into X plus 3 oxygens 3 into minus 2 is equal to overall charge, which is minus 1. By doing this you will get plus 5. So oxygen state of nitrogen plus 5. So zinc, only one element is there, so its ionic charge is the oxidation state. Similarly, if you find nitrogen, you will get plus 3, minus 3 you will get. So now, what are undergoing change? Zinc is undergoing change, nitrogen is undergoing change. So zinc, nitrogen, what is the ratio? Zinc changes how much? 2. Nitrogen changes how much? 5 minus 3. Listen carefully. Sometimes students think that 5 minus 3 change is 2 because they will subtract 5 and 3. But no, 5 and that side 3 is not there, na? Minus 3 is there. So what is the difference? Difference is 8. 5 and minus 3 difference is 8. So 8 you take, and what is the ratio then? 1:4. So zinc items multiply with 4, nitrogen items 1. So zinc items if you multiply with 4, here 4 you are getting, here also zinc item 4. Okay, now charge balance. Okay, left side what is the charge? LHS, what is the charge? Here zinc means neutral state, no charge. NO3 minus 1. Don't take this plus 5. This is oxidation state. Look at only NO3 minus. NO3 minus means what is the negative charge total? Minus 1. Okay. Right side, let's see. Right side, what is the charge? 4 into 4 zinc are there. Each one plus 2. 4 into plus 2 plus 8. And 1 ammonium ion is there. Its charge, overall charge is what? Plus 1. So it is 9. Okay. What is the difference? 9 and minus 1 difference is what? Not 8. 9 minus 1 difference is what? Minus 1. Difference is 10. So we have to add 10. What? H plus or OH minus we have to add? Basic medium means we have to add OH minus. So difference is 10. 10 OH minus we have to add. Which side? I already mentioned previously. OH minus, minus support you are taking means you have to add to the higher side. Okay. So 10 H plus ions we have to add to the higher side. What is the higher side? This is the higher side. Charge is higher here. So this side we have to add. Right side. And how much we have to add? Difference is 10. So 10 OH minus we have to add. That's it. Now answer this question. So before zinc, what is the coefficient? 4. So let's see which is the right option. Zinc coefficient is 4. Nitrate coefficient is what we are getting? 1. Nitrate coefficient is 1. OH minus. Before that what number we are getting OH minus before? 10. So 4, 1, 10. Where is that? Third option. This is the answer for this. Is this understood, everyone? Okay. I'll stop here. And tomorrow we will see another topic, quantum numbers. Quantum numbers we will see. What are those quantum numbers which are important? This topic related to atomic structure. This quantum numbers helps in writing the electronic configuration, which keeps on coming in many chapters in the chemistry. So tomorrow we will see first basics of quantum numbers. Rest 3 days is on quantum numbers only. Tomorrow we will see basics on that. Okay. Thank you. Thank you for listening.
Hi students, hello, good morning. Today our botany class is classification of algae and general characters of bryophytes. What is the main criteria for classification of algae into 3 groups? 3 main classes are there in algae. What kind of criteria is utilized to classify the algae into 3 groups? One is the types of pigments. Second criteria is the stored food, nature of stored food material. Mainly these are two things are used to classify algae into three classes. One is the type of algal pigments, and the second one is the type of stored food material or reserved food material. Okay, such are chlorophyceae, Bryophytes, REDOX. What is the common name of chlorophyceae? Chloro, green algae. Bryophytes, the common name of Bryophytes is the brown algae. REDOX, red algae. Green color causing pigments are more in number or more in quantity. Brown color causing pigments abundantly present. Red color causing pigments are abundantly present in REDOX. What is the pigment that causes for green coloration of the green algae? Especially chlorophyll B, exclusive pigment. What is the pigment that causes for the brown coloration of the brown algae? You learned in last class, fucoxanthin. This fucoxanthin pigment causes for brown coloration for the Bryophytes members or brown algae. Likewise, what kind of pigment leads to the red coloration of the red algae? Phycoerythrin. Phycoerythrin. It is a specialized pigment found in algae only that causes for red coloration. For easy remembering, you have to observe this terminology here. Erythrocytes, red blood cells. Red. Fuco, brown. Chlorophyll B, green coloration. In last class, you started the comparative accounts. Already you made a 3-column table and written a first point, that is the types of pigments. Continue to, and you have to note down the remaining points also. Red algae, brown algae, green algae. REDOX, Bryophytes, chlorophytes. Habitat, marine, seawater, salt water. Most of the green algae are fresh water, chiefly fresh water. Compared to account, second point. Next, coming to the third point. Only few are unicellular, remaining all are multicellular. Only few are unicellular. No unicellular form here. All are multicellular. Every member of the brown algae is a very bigger multicellular. But unicellular members are more in green algae. So in terms of the body size, bigger algae are brown. First place goes to the brown algae. Second place goes to red algae. Least place goes to green algae in terms of the size of body. Okay. Next, third point. Thylakoids are unstacked. Thylakoids are groups. Here also stacked. Stack means group of thylakoids into 2 to 20 thylakoids are there. This point is not much important, negligible point. Next, only is a very, very important point. Fourth point is the most important. Chlorophyll A and D are present. Chlorophyll A and D, A and C, A and B. A is common for all. So the chlorophyll A is the universal pigments. Not only these algal members, even other plant members like Bryophyta, Pterodo, Gymno, Angion, every photosynthetic organism, chlorophyll A must be present. There is no photosynthetic higher plant without chlorophyll A. Except some bacteria, remaining every photosynthetic organism, chlorophyll A must be present. AD combination in red algae, AC combination in brown algae, AB combination found in green algae. Next, exclusive pigment found in red algae only, phycoerythrin. Phycoerythrin. Fucoxanthin, it causes for brown coloration of the brown algae. But fucoxanthin is absent. Fucoxanthin is absent. Phycoerythrins are an example for phycobilins. Here also absent. Here also absent. Another important character, seventh point. Reserve food made of the Floridian starch. The excess amount of food stored in the form of Floridian starch. Whereas in brown algae, laminarin, name of food material. Majority of the remaining algae, green algae, starch. In all higher plants, Bryophyta, Gymno, Angio, all the plants, the excess amount of food material stored in the form of starch. Starch is the reserve food in green algae. Laminar is the form of reserve food material in the brown algae. Chloridian starch is the form of reserve food material in red algae. Next, another important feature in comparative account, gametes, motile stages means gametes and spores are not observed. It means flagella absent. Put the 5 stars here. It's a very important one. Frequently asked in previous examinations. Flagellas are absent. Minus centim is absent. Flagellas are present. For example, it is the pear-shaped gamete or spore. Laterally, 2 flagellas are there. One flagella is a shorter, another flagella is a longer. You have to draw this diagram. This diagram can explain mainly three things. Number 1, number of flagellas are 2. Number 2, one flagella is shorter, another flagella is longer. Number 3, origin of flagella, lateral side of the spore or gamete. In case of green algae, it is the gamete. From terminal region, flagellas will arise. The number of flagellas may be either 2, both should be equal in length, or 4, or 8. 1, 2, 3, 4, 5, 6, 7, 8. Number of flagellas in the members of the green algae, either 2 or 4 or 8. All arise from one common point, that is the terminal area, and all flagellas are equal in length. All flagellas are equal in length. In case of the brown algae, number of flagella only 2. In the 2, one is very longer, another one is very shorter, and origin point is the lateral, whereas here terminal. It is the terminal position of spore or gamete. It is the lateral side of a gamete or spore. Is the flagella totally absent? In red algae, gametes, spores are non-motile, lack of flagellas and cilia. Both the types of locomotor organs are absent. Okay? Are you getting this idea? And note down, clearly note down about the types of flagellas in gametes and spores. Flagellas totally absent in red algae. Okay? In brown algae, only 2, such are unequal in length and laterally borne. Next, whereas in red algae, all flagella, the number is whatever may be, either 2 or 4 or 8, equal in length, and all arise from one common point, that is the terminal area. Terminal flagellas, green algae. Lateral flagellas, brown algae. Flagella absent, completely absent, red algae. Can I change the slide? Try to answer for this model questions. Lichens are algae association with, in lichen is an example for symbiotic organism. The symbiotic association is found between algae and what is the second partner? Yes, good, fungi. Fungi. Right answer, second option, fungi. Good. Next, one more question. Chlamydomonas is a. Chlamydomonas. It is one of the example for the green algae. But green algae is an example for unicellular non-motile or unicellular motile or multicellular colonial form or multicellular filamentous form. Unicellular and motile. It is the Chlamydomonas structure. Single organism. It has 2 flagella. Both are equal in length or terminal in position. Entire this structure is the one cell. Inside of it is one cup-shaped chloroplast is there. Cup-shaped single chloroplast found in Chlamydomonas. Next question. Question number 3. Colonial forms of algae is. What algae is an example for colony form? Chlamydomonas unicellular. What about the colonial form? Yes, third option. Volvox and Idorina. 2 examples are there. One is the Volvox, second one is the Idorina. Both are example for colonial forms. Next question. In algae, the form of reserve food material. What is the common form of reserve food material in algae? Fat or starch or sugars or none of the above. Most of the algae, the majority of the algal members, especially green algae, excess amount of food is stored in the form of starch. Yes, good. Second option, right answer. Good. Next question. Isogamous and anisogamous type of reproductions both seen in. Isogamous means two similar gametic fusion. Iso, similar gametes. Aniso, dissimilar gametes. Both the gametes are involved in fusion or similar. In aniso, one is the smaller, another one is the larger gamete. It is aniso. Both the types of gametic fusions are found in Chlamydomonas. Good. Fourth option, right answer. Next, the product of Gracilaria and Gelidium. Gracilaria, Gelidium, both are example for red algae. That product is used in preparation of ice creams. What kind of ingredient of the ice cream causes for the softness or jelliness or deliciousness of the ice cream? That product obtained from Gracilaria and Gelidium. That is also useful for making of nutrient medium to culture microbes in laboratory. Preparation of culture medium, nutrient medium. Iodine, carrageen, algin or agar-agar. Yes, good. Agar-agar. Agar-agar, the kind of polysaccharide material. Edible material. That's why that is used in ice creams and jellies, culture medium of microbes. Culture medium for microbes. Next, one more question. Fusion of two gametes, the similar size is known as. Easiest question. Similar. Similar. Iso. Yes, first option. Next question. Fusion of two gametes dissimilar in size. Dissimilar in size. Aniso. Even in Ugami is also dissimilar, but one thing is here. In Ugami, female gamete is always non-motile. Female gamete is always stationary, means non-motile. Whereas male gamete is mostly motile, sometimes non-motile. It is the Ugami. In case of anisogamy, one should be non-motile, another should be motile. If the male gamete is motile, female non-motile. If the male gamete is non-motile, female gamete should be motile in anisogamous condition. So it is the oogamous condition. A slight little variation is there between anisogamy and ugamy. In ugamy, female gamete is always non-motile. In anisogamy, female gamete may be motile or may not be motile. Okay. Next, one more question. Fusion between one large static female gamete and smaller motile male gamete is termed as. Static means stagnant or non-motile or stationary female gamete. Answer for this question. Static, female gamete is static. Good, ugamy. Here one more new term is there, that is the siphonogamy. Siphonogamy is the kind of ugamy. Siphonogamy is a type of ugamy. Female gamete or egg is a large stationary and embryo sac. One synergid, second synergid, three antipodals, two polar nuclei. Pollen tube carries male gametes because these two male gametes are non-motile. If the tube acts as a carrier for male gametes, tube, English term, technical name of the tube is the siphon. Siphon. If the siphon is involved in gametic fusion and tube acts as a sperm carrier, male gamete carrier, that kind of ugamy is named as siphonogamy. Siphonogamy found in all higher plants, angiosperms. Okay? Are you getting an idea? Next question. What is the form of reserve food in algae? Already this question is repeated. In algae, starch is a common form of reserve food material. Next, one more question. What is the common name of Chlorophyceae members? Chlorophyceae. Chloro, green. Blue-green algae is another name of cyanobacteria. Cyanobacteria. Brown algae, Phaeophyceae. Golden algae, Bacillariophyceae. Green algae, Chlorophyceae. Actually, these two are out of syllabus. Next, Rhodophyceae members are commonly called as Rhodo. Red. So, red algae, third option. Next, one more question. Members of the Phaeophyceae are also called as Pheo. Brown algae. Yes, brown algae. The pigment responsible for coloration of the brown color in brown algae. Important point. Brown coloration of brown algae. Fucoxanthin. Fucoxanthin, second option. But a special chlorophyll pigment found in brown algae, C. but brown coloration of the brown algae caused by fucoxanthin only. Yes. Next question. What is the reserve form of food material in brown algae? In what form food stored in brown algae? Give answers. What is the form of reserve food material in brown algae? Or PFI members of algae? Yes, laminarin. Laminarin. What is algin? Outer layer of the cell wall in brown algae. Outer layer of cell wall. In cell wall, two layers are there. Inner layer cellulosic in all 3 days of algae. Outermost layer, second layer is made up of algin in brown algae. Whereas in red algae, outermost layer is made up of kerragin. So algin, kerragin both are cell wall materials. Fucoxanthin is pigment. Laminarin is food. Laminarin is a form of food. Fucoxanthin is a pigment. Kerragin, algin are cell wall materials. Right answer, second option. Next question. Mannitol found in brown algae is a type of mannitol. Laminarin and mannitol both are types of form of reserve food material. Mannitol and laminarin both are type of reserve food material in brown algae. Not pigment. Mannitol is not a pigment, form of reserve food material. Next question. Somebody asked about the structure in last class. A leaf-like photosynthetic organ found in brown algae. In all brown algal members, body is the large size, such are called kelps. In this kelp, one green color, very expanded leaf-like structure is there. But at base, for fixation purpose, hold fast is there. Hold fast. What about this remaining larger portion? It is frond. Frond is a larger photosynthetical green colored leaf-like structure. This kind of frond commonly found in kelps. One more question. The pigment responsible for the coloration of the red algae. Red. Fucoxanthin causes for brown coloration of the brown algae. Erythro. Red. Laminar is not a pigment, that is the food material. Chlorophyll D is a pigment but doesn't cause the red coloration. Then right answer for it, second option. Yes, good, second option. What is another name of RBC? Red blood cells. Erythrocytes. Erythro, red. That's why the common name of erythrocytes is the RBC, red blood cells. Next, algae completed. Next chapter, next plant group in the plant kingdom, Bryophytes. Bryophytes. In Bryophytes also several plant groups are there, such are liverworts, hornworts, mosses. But mosses are more dominant over the hornworts and liverworts. In algae also 3 classes are there. Let's just you learn, such are green algae, brown algae, red algae. Whereas in Bryophytes, 3 groups such are liverworts, hornworts, mosses. Mosses are more dominant and a larger group among these 3 within a Bryophytes. The common habitat of the Bryophytes, moisture shaded areas where the moisture is there and shade, cool and damp places are there. That is a favorable area for growth of Bryophytes. Cool area and moisture, shady area is essential. And damp area. Damp, cool, shaded, moisture area is essential for growth of bryophytic plants. Hill surface areas in heavy rain falling area like North India and Ooty, Kodaikanals, where the hilly slopes are there and continuous rains, cool, damp area is there. That area is suitable for growth of Bryophytes. Bryophytes are able to grow in both terrestrial habitats, terrestrial means land habitat, and aquatic habitat. To the presence of 2 different types of habitats, Bryophytes are called amphibious plants in the plant kingdom. Amphibious plants. What is the meaning for amphi? Both. Amphi means both. Two types of habitats are there here. One is the terrestrial, another one is aquatic habitats. Hence these are called amphibious plants in plant kingdom. Bryophytes. Bryophytes are also called as the archegoniates because the female reproductive organ or structure is archegonias. Especially in lower plants like Bryophyta and Pteridophyta gymnosperms, female organs are gametangia. Female gametangia also called as archegonia. Male gametangias, male sex organ and 3Ds. These kind of structures first and all from Bryophyte onwards. Hence, Bryophytes are called first archegoniates in plant kingdom. And these are first embryophytes also. First embryophytes, first archegoniates, atracheophytes, non-vascular plants. Non-vascular plants, vascular tissue is absent. Non-flowering plants, cryptogams, you learn in first class introduction part of the plant kingdom. Tell me once again, what is the meaning for the cryptogams? Non-flowering plants, atracheophytes, non-vascular plants, vascular tissues are absent. Embryophytes, embryo formation occurs in their life cycle first time. And female sex organs like archegonia formation also first clearly observed here. That's why these are called first archegoniates. First archegoniates. Okay. Bryophytes are number 1, archegoniates, number 2, embryophytes, number 3, atracheophytes, mean non-vascular plants, and cryptogams, non-flowering plants. These are important features of bryophytes. They are also called as amphibious plants in the plant kingdom. Why the bryophytes are called amphibious plants in plant kingdom? Why these are called as amphibians in plant kingdom? Amphi means both. Aquatic habitat, it is aquatic, and soil area, terrestrial. These are able to survive in both areas. That's why these are amphibious in plant kingdom. They are primitive land plants. It is the complete surface area of the rock in hilly zones. The first occupants in these hilly areas are bryophytes. Whenever bryophytes are continually grown up to 100 years in this area, then this area gradually make a favorable for growth of other plant groups like pteridophytes, gymnosperms and angiosperms. So these are bryophytes are the pioneers in rocky areas, and these are called primitive land plants. Very, very important point. Primitive land plants are first true land plants. First land plants. Not true, but first land plants are primitive land plants are bryophytes. The plant body of the bryophyte is the more differentiable than that of algae. It is one of the simplest comparison between bryo and algae. Algae is a very simpler. Algal plant body is a very, very simpler. But Bryophyta body is the simplest. Lesser simpler when compared to the algal. Slightly differentiable. Observe here. It is one of the typical moss plant body. It is the gametophytic generation. Haploid plant body. Haploid N. The plant body differentiable into 3 structures. At the base, root-like structures are there. Not original root, just root-like. Here, leaf-like structures are there, not original leaves, just like only. A stalk stem-like structure is also there. Stem-like, leaf-like, root-like structures are present. The leaf-like structures of the bryophytes are called phylloids. Phylloids. Stem-like structure is the colloid. Root-like structures are rhizoid. Rhizoids. All these are technical names. Root, stem, leaf, English terms. The Greek words are technical names of the root, rhizo, stem, collo, leaf, phyllo, seed, spermo, fruit, carpo, flower, antho, flower, antho. All these are English terms, but these are technical names. Oid, O-I-D, oid, like, similar but not exactly equal. Another name of the oid is the pseudo, not original, seems to be original, but not really original. Root-like, rhizoids, stem-like, colloids, leaf-like, phylloids are found in plant body, that is the gametophyte of moss plants. This the gametophyte is a dominant and green color photosynthetic. Provide nourishment for the sporophyte. This gametophyte provides shelter for the sporophyte and provide water and food material for the sporophyte. Sporophyte is differentiable into three parts. Basal food, second part middle elongated structure is there, it is called as the stalk. Third, highly important part for the sake of reproduction, that is the capsule. As in gametophyte, sporophyte also three parts are there. Name the three parts of the gametophyte: root-like, stem-like, leaf-like structures. Whereas in sporophyte, basal foot is there, middle elongated part is the stalk, terminal fertile spore producing portion is the capsule. Okay. One of the similarity between gametophytes and sporophytes. Gametophyte differentiable into three parts, but it is the photosynthetic. Sporophyte also differentiable into three parts, but non-sporophytic. Gametophyte is independent and dominant. Sporophyte is the dependent on gametophyte. It is a non-photosynthetic area, unable to survive its own. It depending on gametophytic generation only. Whereas in higher plants, sporophyte is dominant. Food gradually become as the root system. Stalk become as gradually shoot system, and capsules turn into leaves and flowers. Leaves and flowers. This is about basic structures of or elementary knowledge of most of the moss plants. Mosses are one of the group of Bryophyta. They lack true roots, true stems, true leaves all are absent. In presence of true root-like structures are the rhizoids, leaf-like phylloids, stem-like colloids. This is the plant body. Green color photosynthetic. It is also part of the plant body, that is sporophyte. The major variation between gametophyte and sporophyte. Sporophyte is in diploid condition, gametophyte is in haploid condition. Sporophyte is independent and dominant. Independent and dominant in life cycle. Whereas the sporophyte is dependent, grown as a parasite and reduced. You should note down these two important points. How can you differentiate the gametophyte from sporophyte in moss plants or bryophytes? Gametophyte is independent, dominant, photosynthetic. Sporophyte is dependent and reduced. Grown as a sporophyte, grown as a parasite on gametophyte. Gametophyte provide both shelter and food for the sporophyte. The main plant body is the bryophytes haploid. And in this bryophyta plants, only one kind of tissue is there, parenchyma. There is no sclerenchyma, no collenchyma. Vascular tissues like xylem and phloem also absent. Another important things, the male sex organs in bryophytes is the antheridium. Female sex organ or female gametangia is the archegonia. Antheridia, archegonia. Arche, female. Anthro or andro, male. Andro, male. Main functioning of antheridium is production of anthrozoids. Anthrozoid is the sperm with flagellas, and that is called anthrozoid. But archegonia can produce a single egg, only one egg is there. Single egg, that is a non-motile egg. Egg is the single and non-motile. These are the archegonias, these are antheridias. Antheridias are male organs, archegonias are female organs, nothing but gametangias. Gamete producing sacs are called gametangias. Where water is essential for transmission of male gametes towards the female gametangia. Then that kind of gametic fusion is called as oogamy. In algae, isogamy, anisogamy, oogamy, three are possible. Whereas in bryophytes, only one kind of gametic fusion is there, that is the oogamy only. Female gamete is the egg, larger. Male gamete is the very smaller with flagellated. It is also called as the sperm or anthrozoid or motile. Give rise to zygote. The result of fertilization, this kind of fertilization is called oogamy. Zygote is the resultant of oogamy. In all higher plants, not only Bryophyta, even Pteridophyta, Gymno, Angio, the same kind of gametic fusion is there. One peculiar feature of Bryophytes, zygote do not undergo meiosis. It undergo mitosis only. Give rise to embryo. First time in plant kingdom, and these are called embryophytes. First embryophytes in entire plant kingdom, bryophytes. First embryophytes in entire plant kingdom, first embryophytes are bryophytes. These are the moss plants where the fully moisture, shady locality, cool and damp soil is available. That area is the vigorous growth of bryophytes. All these are moss plants or bryophytes. These are very small, thin-like plants. Appears like a green color carpet on surface of the soil. Green color carpet on soil surface. These are moss plants. In life cycle of the moss plants, first one juvenile stage is present, that is called protonema. It appears like a filamentous. Nema, filament. Proto, first formed phase is the filamentous. That is meaning for protonema. It records their ancestral features. Who are the ancestors of the bryophytes? Algae. In majority of the algae are filamentous structures. In majority of the algae, body appears like a filamentous. So bryophytes ancestors algae. As algae are ancestors of bryophytes. What are the ancestral features recalled by the bryophytes? Filamental structures. Filamentous nature. That is a creeping on ground, green in color, branched and frequently filamentous structures. Second stage, that protonema give rise to adult leafy stage. It is the leafy forms. All these are leafy forms obtained from protonemal stage. Protonema. It is the first structure. Second structure is the leafy form. Protonema give rise to leafy forms. That is the leafy form. Here some filament-like structures are appear. It is the protonema. Proto, first formed filamental stage is the protonema. Next, adult form is the leafy form. We can make some questions here. What is the name of juvenile stage of the life cycle in bryophytes? Protonema. Name of adult stage, leafy form. Try to give answer for this question. Mosses possess root-like rhizoids. What about the phylloids? Leaf-like. Colloids, stem-like. Root-like rhizoids. All bryophytes are homosporous. One more important point. All the spores obtained from a plant body are same kind. That is the meaning for homosporous nature. Homosporous nature. Okay, bryophytes completed. Next chapter is the pteridophytes. I will start in next class. Okay, and bye. We will meet tomorrow.
Yes, good morning, students. I've got a question for you. Very simple question. So define this term. Guys, try defining this. Health. Healthy. What do you understand just by listening, just by thinking about this English term? Health. What do you mean by having a healthy life? Healthy diet, healthy habits, healthy thoughts, healthy body. The word healthy is applied at many places. Healthy environment. Health means, simply speaking, everything happening normally. If all your body cells, tissues, organ system act, behave normally, which means your metabolism is normal, not low, not high. So cells are getting dead and these dead cells get replaced at normal velocity, normal pace. Your hormones are acting at normal pace. Enzymes are acting in given amount of time, normal time. So from day 1 to day 80, 85, 90 years, you had a very, very normal body life, body tissues, organ systems. That was about your body. And healthy means, again, having normal thoughts. Just normal person, just a common person. Not too low, not too high. Just a normal, mediocrity. If you are able to maintain your internal body systems normally, if you are able to maintain external communications also normally, if your socio-economic status was also usually normal, if your psychological state was also normalized, then the state of normality, being normal, is actually considered as being healthy. Healthy means, guys, physically, mentally, socially, psychologically fit. Being physical, mental, socio-economical fit is considered as health. When each and everything is happening proper, having healthy body, healthy relationships, healthy economic status, healthy economy, healthy financial service, and healthy way of leading life, normal, is healthy. If you speak about just internal body health, having a physically fit body with normal mental psychological thoughts could be considered as a healthy body or a healthy person. Guys, if you're healthy, your enzymes and hormones are functioning at normal rate, normal pace, normal speed, velocity, it will be very, very easy for you to perform any task, any work, task assigned to you will be completed very soon because you're healthy. When you're healthy, everything seems to be easy. When you're healthy, each and every task assigned to you will be easily carried by you. You have an ease in doing things. Ease, very easily. But a cell, some cells, or some part of tissue, or a corner of an organ, or some middleman in organ system, if any of your organ system component fails, get disturbed, cannot function properly, permanently or temporarily. Telling again, if your cells, tissues, organs or organ system do not function properly, it might be temporary or permanent. For that amount of time, this ease in performing a task will be temporarily or permanently lost. A task which was easy for you today may become difficult for you tomorrow if it is temporary. But if it is permanent, the task which is easy for you today will become permanently difficult for you for your entire lifespan. So any disturbance in your health, any disturbance in your body systems can impair, can disturb your ease in doing things. Loss of ease losing your ease in doing some tasks, lose in functioning, lost in disturbance in your enzymes, hormones, body systems is disturbance in ease. That was dis-ease. It is neither Latin nor Greek. It was just a simple English. Dis-ease. If you're healthy, easy. Not healthy, dis-easy. Dis-ease. Dis, appear, disappear. Like this, opposites. Healthy is, unhealthy dis-ease. So opposite of healthy is unhealthy. If you're healthy, everything is easy for you. But if you're unhealthy, you will be diseased. This disease could be either temporary or permanent also, right? Now the question was that if your body was diseased, listen, guys, if your body has got some disease, first point for you, reason, how did you get disease? How that disease has happened to you? Is it because of some pollution? Is it because of a pathogen? Is it because of some contamination? Or is it given to you all of a sudden by some radiation mutations? Or unlucky if you are getting this disease condition from your parents, genetical, or this disease might be because of sudden abortion of your enzymatic functions. It might be because of increase or decrease in your hormone levels also. There were many reasons to cause a disease in your body. So first thing is to find out the source of the disease. Is it a pollutant, pathogen, contamination, radiation, mutations? Okay, is it genetical from parents? Or it might be because of any disturbance in your metabolism, because of enzymes and hormones, possible. First thing is to find out what was the real cause behind disease. Think first. Second point for you. Is this disease condition temporary or permanent? So temporary or permanent? If it is permanent, question was, why, how come disease become permanent? Guys, it might be permanent because of genetic. If you're getting problematic genes, DNA from your parents, from day one of your birth, we have to live with the disease like Down syndrome, Klinefelter syndrome, Turner syndrome, sickle cell anemia, hemophilia. These genetic diseases are often permanent. Your immune system cannot help you from them because from day one it is with you. Those people do not know what is healthy life. But if your disease was temporary thing, if you have temporary disorder, temporary disease, temporary symptom because of some pathogen or pollutant or some any other agent, hormones, it could be cleared off. Yes, that could be treated, that could be prevented. You need a doctor for that. But the third point was that 99.9% of diseases happening to you because of a pathogen. Listen carefully. 99.9% diseases happening to you because of pathogen. Pathogen could be virus, bacteria, fungi, helminth, protozoans. 99.9% of diseases caused by these pathogens do not require attention of a doctor or any medical professional, because ourselves we do possess an efficient mechanism, an efficient system to tackle these pathogens. So there is a weapon system inside your body which will take care of more than 99% of your body issues. There is a weaponry protective system inside your body which can kill almost all pathogens entering your body. So this special system which can kill any pathogen usually which can come in contact with the body was referred as immune system. And for information, our immune system can kill any type of non-human cell coming into your body. Immune system can kill all viruses, all fungi, all plant, all helminth, all protozoans. There is no exception for pathogen. Every pathogen could be killed by immune system. Yes, every pathogen can be killed by immune system. That was true. Now you may ask me, if immune system is so strong, if your immune system is so sophisticated and very developed, how come people are getting diseases? Yes, that was a valid question. Guys, immune system has got enough potential to kill all pathogens. It can kill all pathogens provided system can see them. If pathogen comes in contact with immune system, definitely it will be killed. But there are many pathogens which hide away from our immune system. Like HIV, viruses are obligatory. They should live inside your cell. Once virus enters your body, if it is at least for 20-30 seconds in your body plasma, immune system can get activated and kill it. But viruses have evolved a mechanism in which as soon as they come into your body, they will target a cell and enter that cell. When virus has entered inside a cell, virus has become invisible for our immune system, isn't it? If pathogen became invisible, even if you have a very strong immune cell, it is almost impossible for immune cell to kill a pathogen now. Yes, but if you are lucky enough, if your immune system can recognize this pathogen even before it entered a cell, lucky, your immune system will kill that pathogen. And the beauty was that not only killing this pathogen, your immune system will also remember what is this pathogen. It has a memory. We call that as immunological memory. If the second pathogen comes again second time, it will not take much time now. If first response exhibited for one day, if it took almost 1-2 days to exhibit a response the first time, if the same pathogen comes second time again, it will just take 1-2 hours only. So primary response was slow, but secondary response was very fast, very rapid, like this. Okay, so what was the system? Immune system. So whenever we discuss about health and disease, we need to discuss about these three things. What are these? Source of the disease, how disease has happened. Is it temporary or permanent? Permanent diseases, no cure, no treatment possibly. Temporary diseases, yes, we can try. And how does your immune system take care of your body and keep yourself safe and away from pathogens. Done. Study of diseases, their causative organisms, symptoms, and possible treatment regimen. What do you call this branch of biology, branch of medicine, which is concerned with study of diseases, this causative agents, symptoms, and possible treatments? What do you call this? This branch of biology, this branch of medicine, which is associated with study of diseases, what do you call this? That was pathology. Logos, study. Pathos. The word pathos means pain. And word pathos was derived from a term called as pati. Pati means pathogen. Pathos, pain. Study of pathos, study of pain caused by different pathogens is pathology. A doctor who deals with this is pathologist. Pathology lab. Pathology is one of the ancient. Pathologist was one of the first doctors to ever exist. Once upon a time, doctors used to treat symptoms. It was continuing still now, but now the trend has been shifted. Instead of treating a symptom, medicine has advanced in such a way that doctors were now treating pathogens directly. If you kill a pathogen, you may decrease the symptom. But if you just treat a symptom, possibly it may not kill a pathogen. Example, it is very common to you. Listen to this carefully, guys. This information is very much crucial for you and your parents also. Listen to this carefully. You got fever, temperature raised to almost 101, 102 degrees Fahrenheit, not Celsius. 98.6 was Fahrenheit. Okay. So, because of increase in the temperature all of a sudden, your body enzymes, membrane proteins, hormones may not function properly for some time. Make it 1 to 2 days, 3 days at least. Because of increased body temperature, these proteinaceous components like enzymes, proteins, receptors may be denatured. So you need new set of enzymes, hormones, proteins again. It will take some 2 to 3 days time, right? For this 2 to 3 days, your digestion, respiration, communication will be temporarily disturbed, isn't it? What do you call this temporary disturbance in your body system because of increased body temperature? Name this. Because of increased elevated body temperature, you are not able to perform tasks properly. What do you refer this symptom as? Try this. Due to increase in body temperature, your ability to perform some functions was temporarily lost. What is the symptom? Yes, feverish, fever. Correct. Whenever there is increase in body temperature, observe this. Definitely there will be increase in mucus in your respiratory system also, isn't it? A person who is having a fever possibly may also have issues with their mucus in their nose and throat, isn't it? Now, guys, fever is symptom, not a disease. Symptom. So why fever was caused in normal language? If a bacteria entered, possibly if some bacteria entered your body, if some bacteria entered your body, guys, listen to this carefully, it's very useful for you. If a pathogen, bacteria entered your body will automatically increase body temperature, assuming that this bacteria might be killed at increased temperature, because bacteria likes 37 degrees Celsius. So body will automatically feel that if I increase this to 40 degrees Celsius, if I from 98.6, if I move to 99, 100, 101, 102 degrees Fahrenheit, because of increase in body temperature, body will assume that bacteria might be killed. Yes, possibly. What was the side effect? Along with bacteria, our own body cells will also be disturbed, isn't it? So whenever you get fever, you should assume that bacteria or some pathogen has entered body. To kill this pathogen, body hypothalamus has automatically increased temperature. So if you get fever, a good doctor will never ask you to take a medicine, because fever is a response or a symptom to kill a pathogen. Body is trying to kill a pathogen by increasing body temperature. So fever was supposed to be natural, maintained at least 2-3 days normally, because your body is taking time to kill that bacteria. Done? So if you get fever, be with it. Be with the fever, because body is trying to communicate with you. Please don't do any task for 2 to 3 days, because body got infected with pathogen. So first kill this pathogen, first save your body, then you can save time later. First save your body. That was the intention of your body system. But what do? You got fever because there entered some bacteria into your body. What do? Immediately we eat a tablet, we drink a syrup, possibly consisting of aspirin, paracetamol, Dolos, paracetamol. If you take a tablet, medicine, pill, this is a pill which can decrease body temperature. Any medicine which can decrease body temperature is called as antipyretic. It decreases temperature. So you took a pill, paracetamol, and this pill will decrease your body temperature. Yes, fever symptom got decreased, but this pill did not kill this pathogen, isn't it? No. You took a pill which decreased temperature but did not kill a pathogen. Your fever got cured within 1 day, but wait for 1 more day. Again, bacteria still living there. It will again take its function. I mean to say that if you take a paracetamol pill, it will decrease temperature and it will actually is destroying your body's ability to kill bacteria by increasing temperature. No, you should not take pill now. If you take a medicine now, it will decrease temperature. Your pill is telling to hypothalamus, decrease temperature. Hypothalamus is telling to your body that we should increase temperature so that we can kill a bacteria. Take a pill, decrease temperature, fever got cured, but bacteria still lived. Now this bacteria got a chance to live in your body now because you gave it chance by taking a pill. Body is trying to increase temperature to kill bacteria. You take pill and decrease temperature, and bacteria has got an advantage. Okay, temperature got decreased, happy now. This bacteria will now very comfortably go to their target location, enter it, make some changes in body cells and tissues, and will come out with even more dangerous disease. Isn't it? Now guys, tell me, taking a medicine, paracetamol, as soon as you get fever, is it advisable? Yes or no? Do you have to eat medicine if you get fever? Think of it. Are you trying to say that we take pills to decrease fever? No. You're actually ruining, disturbing, damaging your body's immune system. Refer to any immunology book. Ask any doctor, any pathologist, any biochemist, he will tell you the truth. Pills are not supposed to be eaten when you get fever. So what can, yes, Avinash, then what, sir, are you saying to avoid taking pills? Yes, Avinash. Take pills for other body pains like this, but not for fever. Nothing was, you can take pill, but not a pill made up of paracetamol. You should take a pill which consists of antibiotic. Target the bacteria, not symptom. If you take paracetamol, it will decrease symptoms. If you take antibiotic, it will kill bacteria. Therefore, symptoms will be automatically decreased. Which is better? Taking paracetamol pill or antibiotic pill? Decide. Which is better, to take paracetamol or antibiotic? Then why all doctors advise to take pills, sir? Not all doctors, some. Ratna Bujji Kuchipudi. There are many good doctors in society who will never ask you to take pills as soon as you get any fever or symptoms. They'll ask you to stay. Yes, very good. Antibiotic should be the answer. You should take a pill which consists of antibiotic which can kill your pathogen. But the thing was, if you get virus now in body, to kill this virus also, your body has increased temperature, you got fever. Now tell me, guys, you got virus, you got fever, will you take pill now? Yes, Chetan, sir, what if fever lasts long? Chetan, first find out cause of your fever. Is it because of a virus, bacteria, protozoan, or some helminth or HIV? You should know that first. Get to know the reason, you can solve it. Without having a reason, there is no point in taking pills. So now tell me, if you get virus, which pill should you take? Will you take paracetamol pill or antibiotic pill? So good antibiotic depends on bacteria. There are many antibiotics. Many antibiotics are there. We cannot say best one because target. In market, yes, I can suggest some penicillin, ampicillin, erythromycin, chloramphenicol, tetracycline, some groups. Bacitracin. If you take paracetamol, fever got decreased, but virus still lives. Even if you take antibiotics kill bacteria, not viruses. If you take antibiotic, it is waste of time because antibiotics cannot kill virus. Waste. If you take paracetamol or antibiotic tablets, pills, that will not help you to kill this pathogen. What should you take? Antivirals. You should ingest antiviral drugs. They will possibly kill viruses. If virus was killed, definitely symptoms caused because of this viruses will also be decreased. Medicine or treatment is something which you take after symptom. Pathogen enters your body, you should kill it. That is cure, treatment. Vaccine was something, we take vaccine 99%. We take a vaccine before a pathogen actually entered your body. So vaccine was a anticipation, waiting for a pathogen. You took a vaccine, because of vaccine, your immune system got activated. When pathogen really comes now, this activated immune system will now kill the pathogen. Very easy, right? So vaccines prevent disease and symptoms. Vaccine prevents disease and symptoms, whereas medicine will cure disease and symptoms. Which is better? Prevention is better than cure, or cure is better than prevention? Which is better? Vaccine or a medicine? Vaccine or a pill? According to you. Paracetamol is a drug which can decrease your body temperature, and it will be used if there is any disturbance in body systems which gave you fever. Paracetamol is something which actually should not be used. I do not suggest. And if you get a paracetamol, the thing was, you eat paracetamol or not, that doesn't have anything to do with immune system actually. Paracetamol, if you eat paracetamol, your immune system will kill bacteria or not also. So paracetamol is temporary aid only. I mean, it will temporarily decrease your temperature. That's it. Nothing else. There is no clinical significance for paracetamol. It has no effect on your pathogens. Digestive system becomes weak, so we don't take rice during fever. Yes. Digestive system weak. Why do we get weak digestive system? Because digestive system works because on by digestive enzymes. Enzymes are proteins. They will be denatured at high temperatures. How can you digest possibly then? If you have a decrease in enzyme content, you cannot digest properly. That's why doctors advise you to take simple food without much load on stomach or intestine. Take very simple food which needs no further digestion. Glucose is simple food which doesn't need digestion. It will be directly absorbed in the intestines. Whenever you got any pathogen, you should always take simple food because you should not increase load on your digestive system. That was the logic. Soups, eggs. Vaccine was more preferred than medicine. There are many vaccines against rabies. Viral vaccines were there, bacterial vaccines were there, protozoal vaccines were there. Still science was getting advanced day by day. Scientists were creating more and more vaccines. Scientists were trying to get as many vaccines as possible so that nobody will suffer from any pathogen in future. Hope scientists get succeeded in discovering, inventing, manufacturing, creating more vaccines. The thing was, you can never get rid of pathogens. Your mouth right now, our mouth right now has 10,000 bacteria at least. So this room where we stay, it has more than 10 millions of bacteria. There is one person standing here among 10 million bacteria. So 1:10 million. This world belongs to Monirans. This world doesn't belong to us. We are living among bacteria. Bacteria are not living among us. This world belongs to prokaryote. They came first. They live forever. They will kill us. As you have got conscious enough to say we dominate. No, we do not dominate at all. Among 5-6 kingdoms you have, the most dominating kingdom was always Monera. Monera can kill you like a parasite. Even if you die, Monera will decompose you. It will not leave. And Monerans have immortal lifespan. It is physiologically, metabolically impossible to kill a bacteria. You know why? They have circular DNA. There is no death for bacteria. This world belongs to them. So, as we increase vaccines and medicines, these viruses, bacteria, fungus will have more challenge. They get selected by nature now. So medicine has to progress continuously because you got more and more bacteria coming to infect us. It will never stop. It is a constant fight between pathogens and vaccines, pathogens and medicines. Hope some of you invent more vaccines to save all of us. Possibly, right? So the point of today's discussion is to let you know importance of a pill, immune system, vaccine and medicine. Yes, understood. So please be very careful with vaccines, medicines, pathogens, symptoms. Treat disease, not symptom. If you treat symptom, it is temporary. If you treat bacteria pathogen, it is permanent. Understood? Okay. So we'll sign off at this moment. We'll meet again once you come here. Take care. All of you, stay hydrated, stay home. Okay. Thank you.